It is appropriate to test hypotheses in science and even theories. And so there are three models on how life's diversity reached its current point: the evolution model, the creation model, and the intelligent design model. And one can test the predictions of these models using genetic analyses, such as an analysis of the transcription factor HOXA1. Transcription factors are those proteins which bind DNA and thus regulate gene expression, deciding which genes get expressed, turned on, and this group is critical during embryonic development. Embryonic cells have all of the genes of the human genome, but to become part of the brain or the liver or this part of the limb or this part of the neural tube, etc., they must express not all genes, but the appropriate ones. And so the process of differentiation requires transcription factors which guide which genes are turned on. Which genes help a human embryo become human? Are these genes unique to humans? No. A subfamily of the helix turn helix transcription factors, which date back to bacteria, is known as the homeodomain family. And the genes in this group underwent frequent duplications, allowing the homeodomain family to amplify as time went on. This group was originally described in Drosophila, and so the genes which help humans develop into humans are not unique to humans. Once again, homeodomains were first discovered in Drosophila. And the duplications formed a nested hierarchy distribution similar to the predictions of the evolution model. The creation model holds that all organisms outside of kinds are 100% unrelated to each other and equally unrelated to each other. So certainly the existence of homeodomains and the distribution of related homeodomains does not match its predictions. And then finally, in the intelligent design model, the idea of irreducible complexity simply does not apply because homeodomain genes were slowly duplicated and modified throughout the evolutionary lineages. One group, particularly the homeodomain clusters or the HOX cluster genes, were important in giving embryonic cells positional information in an axis. And so the order of the genes in the chromosome actually predict the expression of these genes in cells in the same linear order along axes in the developing embryo. Duplications allowed the one HOX cluster present in invertebrates to become four HOX clusters in the chordates. And it was these separate four HOX clusters which have allowed human embryos to get a great deal of positional information and develop complex structures. In addition, if one were to sequence the HOXA1 gene, not only do other vertebrates possess this gene, but the sequence comparisons then show that sequences reflect the nested hierarchy of relationships that other anatomical and genetic studies have shown. So note that instead of being 100% unrelated, the sequences of apes, like chimpanzees, gorillas, and gibbons, are close to 100% identical to the sequence of a protein which helps human embryos develop into humans.